So I think that if we can start, okay, the reason that Dr. Salvador Macip is with us is to talk about his science, his research. And Dr. Salvador Macip, he was born here in Catalonia. He's a physician, a researcher, and as you can see, a little bit of a writer. Writer in discussing in a lay language biology and medicine. He got his degree in medicine at the University of Barcelona, where he also got his PhD in human physiology. From there, he moved to the Mount Sinai in New York, where he stayed for 9 years. And from there, in 2008, he moved as group leader in cancer and aging at the University of Leicester in United Kingdom, where he's also professor of molecular and cellular biology. He's also a professor in the Universitat Oberta de Catalunya here. So he has this double affiliation. He has a deep interest in biology of leukemia and senescence and aging. And in fact, today's lecture is about resistance and relapse in B-cell malignancies. And this tale, a tale, like in the literature, of mutation, oxidation, and senescence. And Salvador? Please remember, you can ask questions using the online system that we have.
Thanks, Manel, and thank you for inviting me to give this talk. And before we start, please buy the book. We need to pay our bills. And today, I thought I was going to tell you a bit about the research we do in my lab. In my lab, we work mostly on cancer and aging, and the crosstalk between cancer and aging. And since this audience is more, I guess, actually, this disclaimer should be that you probably know more than me about leukemia. I'm a bit new in the field. I have been working on leukemia for around 10 years or so. And I wanted to show you some of the lines that we've been doing in the lab in these past years and what we're trying to achieve right now. Mostly, I think our goal or our interest is in trying to understand why some B-cell malignancies are resistant to treatment and why some eventually relapse. And we think, or one of the things that we want to propose is that there's obviously many factors defining this, but mutations are the ones that we already know, but oxidation senescence may be playing a role as well in this process. And this link, senescence, is the thing that would link the aging side of the lab with the leukemia side of the lab, and I'll bring this back together at the end of the talk. So just to get started, let's see, am I doing it right? Yeah. So I found this rather cryptic graphic on the internet the other day when I was looking for personalized medicine. Our goal is to find our goal, and I probably could include you all, is to try to find better therapies for B-cell malignant, for leukemia, lymphomas. In our case, we're mostly focusing on CLL, DLBCL. We've done a bit of HCL and other B-cell malignancies. And one of the current treatments and future treatments of cancer, leukemia and all cancers, is personalized medicine. And this looks taken from a science fiction book, actually tries to summarize the whole concept of precision medicine or personalized medicine, where you get all this information from all the omics, all the genetics, the genomics, and so on, and to try to see what makes that person unique. That information is processed through a series of algorithms, and eventually the final point would be to choose the right drug for the right person, for the right disease. And I'm not saying anything new. That's sort of the background, the basis of personalized medicine, and that's going to be sort of in the background of my presentation today, how we find better ways of personalizing treatment in B-cell malignancies. We started in this field publishing a small review, this is I think 10, 12 years ago, about what was new in the field and where the field was going for B-cell malignancies in terms of precision medicine. And already at that point there was a big change happening with the approval of the BTK inhibitors, the BCL2 inhibitors, which have completely changed the outlook of these diseases, as you very well know. Now we have a lot of precision medicine, so we have a lot of drugs that target specific proteins, mutations in the cell to treat these B-cell malignancies, but we still have a very heterogeneous response, and relapse and resistance is still a big problem. Those numbers are the 5-year survival rates, which are very good for some of them, like CLL has a very good survival rate, not so good for DLBCL, very bad actually for MCL. And in many cases, after 10 years or so, this disease relapses, and even with the huge effect the Btk inhibitors, BCL2 inhibitors, and the other specific inhibitors have, we still have that issue of why these cells would acquire resistance. The easy answer obviously is mutation. You have the selective pressure on these cells. Eventually, it's going to be a mutation, and we're going to select the clones that are resistant to the drug. But we think there's much more than that, and that's not. It's a much complex picture probably than just that. When we did a quick experiment at the beginning for this paper, actually what we were doing, we had a panel of CLL cells, MCL cells, DLBCL cells from our bank. And what we found is actually, and again, this is no surprise, but at that time it was the time we were, there was all this talk about the heterogeneity of cancers and the heterogeneous response to drugs. The response to targeted drugs is very heterogeneous in these cell lines, and obviously in patients. As you can see here, for instance, in CLL, this is the increase in cell death in apoptosis as you increase the concentrations of this BCL2 inhibitor. And there is an increase very clearly in all the cells in cell death, but as you can see, the difference is very wide, it's very heterogeneous. Some cells would respond very well to this drug, some would not respond that well. And as you go down to other diseases, for instance, the LBCL would not respond at all to this particular drug, and the MCL is even more heterogeneous, the response that you see there. So what we need to try to understand is what makes these cells sensitive to this particular drug. So try to find prognostic markers for resistance and again for relapse of this disease in order to define this personalized therapy. So yes, we have a lot of new targeted therapies coming up, but we still have this issue of this heterogeneous response and this relapse of and resistance in some cases. One of the first papers we published in this field is an example of this personalized medicine. It's a case study we published in the New England Journal a while back about a patient that had refractory hairy cell leukemia. And the particularity was that he had a high cell count in blood. And we did a genetic analysis of that patient, and we found a mutation that at the time was being known that it was quite common in HCL. Now we see that all the classic forms of HCL would have this BRAF V600E mutation. And what we did is one of the things that we've been trying to do in our studies, try to repurpose drugs. So we already have a lot of drugs that work or should work in some cancers. The problem is that we have to find the right targets, the right people to get those drugs. And I think this is one of the key and one of the concepts that I'm going to be talking about today, the whole idea of repurposing drugs. These are drugs that when you try them in the general population, the results may be very heterogeneous and you may get to the conclusion that these drugs don't work. But they do work for a fraction of the patients. The key is try to identify who are these patients that would benefit from a particular drug. In this case, this was very simple. It has a mutation, and we already have a drug for that mutation, a drug that was not given before in HCL. Vemurafenib at the time we were describing this had not been, we were one of the first groups saying that vemurafenib could be a drug that could work in HCL. And as you can see on the left, so the clinical parameters, when we started the treatment in the dotted line, the first graph is the white cell count. And as I was saying, that this patient had a lot of malignant cells in blood, and they dropped immediately when we started treating them with vemurafenib. Again, first time that this was given in patients, we had to, the ethics was complicated for this. We had to apply for this compassionate use of the drugs, and it worked very well. And platelets went up right away. The malignant cells went down. So it was an example. As you can see at the top, you see the kinase assay for the BRAF, and it really went down as it should after treatment. So this is an example of how personalized medicine works very well. So we can look for mutations, and if we already have drugs for that mutation, even if they have not been given before for that disease, that's something that we can propose to use. Problem is that in this case we saw resistance and we saw relapse. So this is a success story. It was very nice when we published it. The patient was very well, was alive. Actually, when we started giving the drug, this patient was about to die, and he was already refractory to all the treatments, and he was in a very poor condition. We gave him the drug. He was very fine for a few months, and then again resistance and relapse, and that's one of the issues. So we have a very specific, very targeted drug, very as much personalized medicine as you can have, but that again puts you into this position that resistance and relapse is even more common when the more targeted and the more personalized the treatment is. Again, how we solve that, and I don't have the answer for that yet, but that's one of the things that definitely we try to investigate, how you can prevent that resistance, that relapse that happens later. What we've been doing in the lab, I'm going to show you just quickly some key findings of some of the papers that we published in the past years, and some new data as well. One of the things that we try to do to avoid this relapse is combination therapies. And again, probably you are even more aware than me that combination therapies is one of the way forward, not only for leukemia, lymphomas, but for most cancers. Single drug treatment, even with targeted drugs, doesn't seem to be the way. Again, because of this relapse that is more common as we go more deep into the personalized and targeted therapies. And what we did here is get a bunch of drugs. I mean, we were encouraged by the success we had with Vemurafenib, and we say, why not do a blind screening of drugs that we have in the lab that we know are can kill cancer cells, but have never been tested. In this case, we did it in CLL. So we did a series of viability assays in different cellular models that we have. We tried a group of drugs, and we identified this CDK inhibitor, dinaciclib, as a drug that had a very potent effect on CLL cells. And that would be the first graph that you see, the cells really going down, this huge amount of cell death happening at very nanomolar concentrations even. And there's different, the first A and B are the screenings of different drugs. B and C are just examples of dinaciclib, different concentrations of dinaciclib in CLLs. We tried this also in patient cells, and we saw the same idea. So what we found here again is a drug that's been proposed to be used in other cancers, never had been checked in CLL. And we say, okay, so CLL cells seem to be particularly sensitive to this drug. What can we do with this? So try to propose combination therapies. Can we use this drug as an extra layer of treatment to avoid this relapse that we see in targeted therapies? So we combined, in this case, things like ABT199, again, BCL2 inhibitors, or Ibrutinib, very good and potent targeted therapies, with a drug that is perhaps more blunt. Dinaciclib is definitely not a targeted drug. It's just, at least not in the case of leukemia, it's just a blunt CDK inhibitor that should work in any sort of cycling cell, like a cancer cell. And when we see the difference in the cell death curves here, is this a quite significant synergistic response when you add very small concentrations. You can see we were using 0.1 micromolar in some of them, or even less in some graphics of dinaciclib, to try to enhance the targeted therapy. So I think, in a way, and this again goes probably for most cancers, cancer treatment has been going in a bit of a pendulum. So we started with the chemotherapy, which is a complete blunt instrument with no targeted anything, just trying to kill cells. Then we went to the other side, just okay, now we have targeted drugs. These are supposed to be the magical bullet, as we saw in the media at the beginning of the century. So we're going to cure cancer by having these sort of very targeted drugs. Now we realize that the more targeted the drugs are, the more resistance we're going to get quickly. And then we're swinging back, I think, to some middle point where we can combine targeted therapies, and definitely there's good examples of this in leukemia treatments. Targeted therapies together with some other drugs that may be classic chemo, or could be things like this, that more blunt drugs like dinaciclib that could potentially enhance or prevent this relapse. Again, these are very initial studies, let's say. So with cell culture models, this would have to be taken forward with other preclinical models to actually validate whether that is something that could work or not. We've been doing this. We did this for a while, just looking for, again, repurposing drugs. We published a few papers on this, like CDC 907, which is a PI3 kinase inhibitor slash has other HDAC inhibitor, other inhibitor properties. In CLL also we found some synergistic effect, and the idea is that these drugs like CUTC or even the MEK, we tried the MAP kinase inhibitors, which were in that case not so useful. Our idea was all the time try to see if we can block pro-survival pathways. So okay, you treat a cancer, and again this will probably work for any cancer. You treat a cancer with a targeted drug, and then you add something that lowers down all the pro-survival signals that would appear in that cell. For instance, blocking MAP kinase pathway, PI3 kinase, or other pathways that could potentially feed back the cell and just bring it back to the relapse. And these are some papers we published with some ideas, at least, that could help potentially design new therapies. One of the things that we worked on as well is the role of p53 in B-cell malignancies. And p53 is my sort of my pet protein. I've been working on p53 for quite a long time, and actually this was my gate of entry into leukemia. This collaboration we had with this clinician Martin Dyer, that he was the one that observed, actually, if you see in the top figure in the top panel, you see how the p53 levels are very high in B cells, especially when you give them the sort of microenvironment they like to thrive, like CD154, IL4, or anti-IgM, or any other sort of thing that kind of mimics the microenvironment of the proliferation centers. And this is very unusual for me, coming from epithelial cancers. I'm mostly been working on epithelial cancers. Seeing that much p53 in a cancer cell, the first thing you think is, okay, this is a mutant p53, because p53 cells would not like p53. As soon as you have p53, the cell will die or arrest or something like that. That is not the case. As you very well know, the mutations in CLL for p53 are quite sort of late events. It's not something that you see very often initially. And in these cell lines and in patient cells that we use, we found that these were wild type p53. And again, a very sort of heterogeneous response. But if you see the levels of p53 in the cells, in patient cells cultured in these conditions, in CD154, IL4, again, to give them the boost to thrive in culture, they would all of them increase p53. And in parallel, they would increase the proliferation. Ki67 would measure proliferation. So this goes against all the dogma of the p53 field. If you have wild type p53, the cell is not going to grow. It's not going to survive. So why is it? What makes these B cells so special that can withstand this much level of p53 without dying? Why they're not dying? Why they're not arresting? Again, there's very heterogeneous response, but most of them would have these high levels of p53 with quite a lot of cell growth. This is what we still trying to understand. In this paper, we found that this p53, one of the reasons we thought, okay, maybe it's there, it's wild type, but it's not working. But it is. This p53 is phosphorylated and it's active. We see phosphorylation at the key residues of p53, and we still see the expression of the protein quite a lot when you block protein formation with cyclohexamide. So we still don't understand at this point. We don't understand why would this cell tolerate this much p53, active p53 on top of that, so phosphorylated and a kind of protein that would be ready to actually kill the cell. Serine 15, for instance, on B is one of the markers of activity of p53. As you can see, 60, 70, 80% of the cells in some patients would have phosphorylated p53 in serine 15. Again, very counterintuitive. These cells should not be growing, should not be alive. We think that what is happening is that in the microenvironment that we create in these cells in culture, again trying to mimic the proliferation centers with CD154, IL4, or other stimulus, what we see is an increase in ROS, in reactive oxygen species. You can see in A, that's a measure by FACS of ROS, and as you can see, there's a huge increase, fold increase in the intracellular levels of ROS. We see it also in B, different kinds of cells of different kinds of culture. You can see increases in ROS. And what we think and what we saw here and with our hypothesis was that this ROS is an oxidative stress. So being in this microenvironment, in these cells, they will be subjected to this oxidative stress. And the cell tries to cope with that. And that's one of the reasons why all these pro-survival pathways may be enhanced in these situations, may be expressed and trying to dampen down the response of p53. So p53 is induced by ROS. When you block ROS with NAC or GSH, which are antioxidants, you obviously see a reduction in ROS and you see a reduction in p53. And we think that what is happening, again, going back to the main point, which is trying to understand why B cells may be more resistant to treatments, more resistant to anything that induces p53. We think that what this is happening, this sort of low level of reactive oxygen species, low but because it's not enough to kill the cell, but it's just is quite enough to increase the reactive oxygen species intracellularly. This achieves perhaps a protective state, makes the cell raise the threshold that needs to actually be killed by a p53 action. And again, there's many drugs, many chemotherapy, the drugs that would act through activation of p53, that in these cells, they would find some sort of a natural resistance already in the cell to apoptotic death by p53, because the cell has through potentially ROS and a higher basal level that gives the cell some resistance and needs forces then to push this level of p53 even higher to actually induce apoptosis. So in other cancers, that's not usual. You would not see that much ROS expressed in cancer cells, and therefore perhaps we're starting in a sort of a lower threshold. And if in those cells are not used to ROS, they're not used to p53, you treat them with chemotherapy, they would just probably die quicker. These ones, B cells, perhaps they have learned to survive and to withstand this higher sort of level of ROS and higher levels of p53, which then makes them put them in this state of sort of resistant to certain drugs. And again, this is not obviously the only explanation, but it's something that we think contributes to the resistance we see in some B cells to actually some of these of these treatments. And this is new data that we're working on now, trying to understand whether there's a link between the antioxidant capacity of the cell and the resistance to drug. Because we found, and this paper was published recently on that when you treat cancer cells, there's always, or most of the time, there's some number of cells that would persist after that would survive therapy. Now, those cells have a particularly high level of ROS, and this was a paper published, I think, in Nature recently as well for epithelial cancers. So this matched our idea of this sort of threshold of ROS, perhaps providing some potential resistance to the cells and resistance to treatment. And what we've been doing lately is studying, and we're studying mostly DLBCL, but also other B-cell malignancies, again, going back into our bank to look for cells. And first of all, we saw in the color graph at the top, you see that the basal level of ROS is very different. It's definitely very heterogeneous in these cells. So some cells have like at least threefold or more, or perhaps even more. Actually, no, yes, a few orders of magnitude more ROS compared to the other cell lines. Is this part of what determines that these cells may be more or less resistant to a particular drug that may be working through that pathway? We have generated these TMD8 cell lines that are resistant to Ibrutinib, and we are comparing now the antioxidant response of the resistant cells compared to the sensitive cells. Mostly also because Ibrutinib, as you probably know, there's an oxidation of BTK in C481 that actually is resistant to the most common BTK inhibitors, the covalent inhibitors like Ibrutinib or Tirabrutinib. So does that have anything to do with the fact that some cells may have higher or lower oxidative levels? Is that helping in any way these cells become resistant, for instance, to BTK inhibitors or perhaps other drugs? This is very much ongoing work right now, and hope to have some more information soon about whether the basal levels of ROS and or the ability of these cells to buffer this ROS plays any role in the resistance to BTK inhibitors or other drugs. And let's talk about BTK. BTK is very well-known protein, and again, probably you know much more about BTK than me in the context of leukemia, and we know that it's upstream of the pathway on BCR and the BCR receptor, and that triggers is part of the kinase that triggers this sort of cascades of events that ends with a lot of things happening in the cell, mostly perturbable pathways like PI3 kinase and so on. Gracias. All right, so we have BTK up there, and again, in this quite very complex pathway. So BTK, obviously, as we saw, is one of the targeted drugs that target, one of the targets that we have now, drugs for that are more successful, the Btk inhibitors in treating B-cell malignancies. And what we found interesting about BTK is that it has a different role, or it plays also a different role that was unexpected. So BTK is part of an oncogenic signal in B cells. So you have, that's why blocking BTK works so well in killing a lot of B cells. But what we found is that BTK induces senescence, and we'll be talking a bit about senescence now. So it has actually a tumor suppressor properties, mostly when we check in epithelial cells. So what we saw is that BTK has these two phases. BTK would phosphorylate a lot of different things. Many of them are proliferation and survival signals, and again, this is important for B-cell malignancies and for the growth of these malignant cells. But BTK also plays a role in the damage response pathway by phosphorylating and blocking MDM2, which is the protein that blocks p53, phosphorylating p53, which activates p53 again, and phosphorylating p73, which is a partner and a family member of p53. Now, this leads to apoptosis and senescence. So BTK is actually important in this context to kill the cell. It does exactly the opposite that it does in B cells. And this was quite a surprise when we found this a few years ago. This is a quick review that we wrote at that time, explained that BTK, we've been trying to inhibit BTK in B cells, but actually inhibiting BTK in epithelial cells may not be that a good idea, because it has a tumor suppressor role there. So what happens also in B cells? That's one of the things we are investigating now. Does BTK also have this sort of good side in B cells? Let's talk a bit about senescence, and we'll bring back again this into the context of resistance and relapse. Senescence is this state, irreversible growth arrest state that happens in cells through different stimulus. One of them is aging. As the cells age, telomere shortening, you've probably heard that, triggers a series of damage responses, leads to senescence, which is this sort of morphologically different kind of a state, as you can see here, in which cells stay there and don't grow back. So we have, if you compare this, for instance, these are epithelial cells, you compare this sort of, I know you can see it from there, but believe me when I say that these cells are sort of small and square. When they become senescent, they become sort of long and flat, and they don't grow. These are cancer cells that are growing. These are cells that are not growing when they become senescent. And this is important, again, it's a tumor suppressor mechanism. It happens in aging by telomere shortening. It happens in response to damage. So it's a way to prevent damaged cell to divide. And what we found basically is that if you use BTK inhibitors, this effect disappears. So when we try to induce senescence, it just doesn't happen. So no BTK, no senescence, at least for the part of senescence that is induced by p53, which is the most important, one of the most important inducers of senescence. So we published this paper showing that basically that p53 activity is very much determined by BTK in terms of both apoptosis and senescence, and that if you block BTK, you block senescence. Don't want to bore you with the details here, but for instance, here you can see cell count. This is a cancer cell growing. If you treat them with ibrutinib, these cells just basically, sorry, the opposite. These cells are senescing. When we induce them to senesce, these cells with ibrutinib don't senesce because they basically bypass the p53 pathway. We did it with different ways of showing this. Basically, everything is basically saying the same thing. So this is actually the beta-gal staining is a marker of senescence. So when you use ibrutinib, there's no induction of senescence in these cells. So basically, what we found here is that BTK is actually a very important part of a tumor suppressor mechanism like senescence, a key tumor suppressor mechanism. This now is going to take us through a slight diversion through another place, but we'll bring it back to leukemia soon. What is the role of senescence in aging? So the question is, why do we age? It's maybe a bit of a philosophical question, but we can see aging, we can describe it very well at the sort of a macroscopic level. We can see what is aging, and we can see what difference between young and old. But the actual reason why we age, if we look deeper, we can see that our organs are aging also at the same time. So we could say that we age because our organs age. So a liver of an old person doesn't look like a liver of a young person for sure. So there is even morphological macroscopic changes in our organs that underpin aging. And even more, if you go even deeper, then we can say that we age because our cells age. And here is where senescence starts playing a role. As you can see, that if you look at the tissues and you start looking at the tissues of all the organs, you would see that as we get older, we start seeing the accumulation of these cells. The senescent cells are in pink here, and there's some when you're young, there's more as we get older, and there's a lot of them when we are really old. And why is that a problem? So the first hypothesis was that these cells had to play a role in aging somehow, but it took a while until perhaps the last 10 years that things started to change, that we saw that senescent cells actually have an effect, and this is the sort of effect they have. So when we're young, all these stresses that I was saying, so like different damages, different things that can mutate the cell, can lead to senescence. These senescent cells here in blue, they would accumulate with time, but they would also secrete something we call SASP, the secretory associated, senescence associated secretory phenotype, basically the things that are secreted like cytokines, chemokines, and different factors that are secreted by senescent cells that actually call the immune system. And this immune system clears the senescent cells from tissues. The tissue regenerates then and stays young. So this is the normal process of what should happen with senescence. As we get older, this starts not working as it should. And certain pathologies, cancer one of them, aging itself leads to actually this bad state in which we have senescent cells not clear, they're stuck in the tissues, the immune system is trying to get rid of them, but it can't, so the macrophages also remain there. That eventually leads to fibrosis. Fibroblasts are called here, and we have this sort of chronic low-level inflammation in tissues that disrupts the tissues, and that's the explanation of why we age, or one of the explanations we think we age. This sort of mess that senescent cells create with time makes this tissue not work at all. If you compare a normal tissue or this tissue that should be able to do whatever function it does, here there's so many things happening that the function starts going down, and that's why we think liver would not work so well, any other, the kidneys would not work so well, the skin wrinkles and so on, just because of that. So there is a bad side of senescence. And this, again, leads to, we think, the changes we see on old age. And also we see that this promotes cancer. So all these factors that these cells generate, if these cells are not cleared, are actually creating an environment in which cancer cells are happy to grow. And we think that that plays a role. Actually, there's a different hypothesis, and we published also a review on that on epithelial cancers. That we think there's a reason that these cells may be playing a role in keeping the cancers relapsing and growing after treatment. Because treatment, chemo, radiotherapy would induce senescence. If this senescence is not cleared, as it happens often after treatment, that would create this sort of microenvironment with a lot of growth factors and things that would actually help induce a relapse. Now, again, I'm talking from the perspective of epithelial cancer or epithelial cells. This is where most of the studies of senescence has been done. Now, does this work also in leukemia? And that's one of the things that we are trying to ask now. Before that, I'll show you the effects of Ibrutinib. So now you've seen Ibrutinib being used as an anti-leukemia, very useful anti-leukemia agent. Now here we're using Ibrutinib in mice, in a mouse model, to actually try to delay aging. So we saw that BTK inhibitors stop senescence. Now, if senescence plays a role in aging, so the next question is, does that work in vivo? Can you give BTK inhibitors in vivo and improve aging? And this is basically what we saw in these animals. So what we here, this is a long paper we published last year, but I'll show you just two things: lifespan and health span. Lifespan is basically how much the animals live. This is the average lifespan of the animals, and as you can see, the animals treated with Ibrutinib from early on would have a longer maximum lifespan, an average lifespan at this point. Now I have to say that these are fast-aging mice. You can see these mice are dead in a much shorter time compared to the actual mouse models that we use. And we use this model because it's cheaper and faster to do experiments. We don't have to wait a couple of years to see the animals survive. So what we saw, and this is again a normal practice in aging research to use these sort of models, at least for the first experiments. And we saw that Ibrutinib actually is prolonging lifespan, but even more importantly, is prolonging health span. So health span would be defined by the amount of years that we spend in health, or the amount of time. And the way we measured it in these mice is with this clinical frailty scores. It's the similar features that you use in humans to assess how frail you are in your old age. And as you can see, the frailty scores are low when the animals are young. As they get older, the control animals quickly deteriorate and score very high in this frailty score. And the ibrutinib treated mice retain these much lower frailty levels. So not only these animals live longer, but they also live better by using ibrutinib. And here's the same data plotted differently. So basically, here's a drug that you guys are used to see used as an anti-cancer agent being used as an anti-aging agent. And that also makes us wonder, what is happening to all the people that are getting ibrutinib or Btk inhibitors in the clinic? Obviously, that's a different situation. We're having patients with cancer. They get the drug, they get better of the cancer. But at some point, something like this may be happening in their tissues. And that's one of the steps we're trying to do now, try to see whether we can get some data from this, almost you could say, clinical study that's already going on with people getting ibrutinib. Is there any improvement in the aging of the tissues in these patients getting ibrutinib, apart from the effects that you would see against malignant B cells? Are any benefits from not having senescent cells or not having so many senescent cells in the tissues in terms of aging? And in this paper, actually, we showed an improvement in cognitive aging. So the cognitive functions were improved in these mice. We are about to publish a paper on muscle, and we're getting ready a paper on the heart. So at least brain, heart, and muscle are much protected against aging when you give them ibrutinib. Is this happening in humans as well? And another flip side of this story would be, so if we say that senescence is a tumor suppressor mechanism and BTK blocks p53 and senescence, would ibrutinib lead to secondary tumors? And we've been trying to scout the publications on secondary tumors in ibrutinib treated patients. And there's not so much information, perhaps because there's not long enough follow-up of this patient. There's some reports of secondary tumors in patients treated with ibrutinib, but you don't know what's the cause of that, whether it's coming from somewhere else or whether ibrutinib had any role by blocking basically p53 and enhancing the chances of these people having other types of tumor. Again, this is something that, especially for you guys working on leukemia, something to think about. What are we doing with ibrutinib? See, if this is something that actually is blocking a tumor suppressor mechanism, is blocking p53, is that going to give us problems down the line? So is it saving patients because it's reducing B cell burden, but is at the same time blocking p53 somewhere else? Is that going to give us problems, or is that going to give us some extra benefits that we don't know? For instance, in reducing aging. Again, a lot of open questions, and again, an example of how the many phases of many proteins. So we like to think of proteins in a pathway that is always linear. So this protein induces this protein, and this has that effect. But as you know, in real life, in biology, this thing is much more complex. Everything works in networks. So ibrutinib is involved in many, obviously by phosphorylating so many things, it's involved in many pathways. And here's at least is something that is interesting would be interesting to follow up and see what happens when we're giving ibrutinib to people in terms of aging and in terms of tumor suppressor mechanisms. Going back to leukemia now, we just said that the presence of senescent cells would be something bad for the microenvironment in epithelial cancer. Would be that the same in lymphomas or in leukemias in the microenvironment? And what we're trying to do now is to see if senescence is contributing to B cell resistance. We know it does in epithelial cancers, but does that play any role in B cell malignancies, or is the architecture of B cells just not something that would be influenced by the presence of senescent cells around them? So the first thing we're trying to do is culturing different cells and treating them with, again, this is from our library of B cell malignancies, a lot of the LBCL cells, trying to see if what's the concentration of different drugs that we've been studying in these cells, what concentration would induce senescence, and whether that presence of senescence is proportional or has anything to do with the resistance or the sensitivity of a particular cell line against a particular drug. So again, trying to link, and this is, as I was saying, this is a work in progress, and I don't have the answer for this yet, but we're trying to see whether senescence, which we know is happening, we see here some examples of senescent cells after being treated with some of these drugs. And they don't look the same as they look in epithelial cells, but you would see some sort of a blue staining that is the staining that indicates presence of senescence. So we know that senescence can happen in B cells. That's been published before, and we are showing it again in response to some of these drugs. So is the amount of senescent cells we see in response to this in any way proportional to the resistance, or does it have anything to do or not? That's one of the questions we're trying to answer at the moment. And that would be the last bit of my talk, is one of the things we want to do. If senescent cells are so bad, is there a way to get rid of senescence? So you saw how we use ibrutinib to prevent senescence, but this, as I said, could have all these side effects of perhaps blocking p53 and perhaps promoting cancer. Would that be a problem? Can we do it after the senescent cells show up? So if you remember the figure I showed you before, the problem is not so much the emergence of senescent cells, it's the fact that they're not being cleared from the tissues. So is there a way to specifically target senescence? And we're going back to targeted drugs. And here we're talking about something that we call senolytics. Senolytics are drugs that kill specifically senescent cells. There's some chemicals that do that, but again, these are sort of blunt tools that are not very specific. Now we're some groups, my group and other groups, are trying to work and design what we call targeted senolytics. And the first paper we published on this is using nanoparticles, which is a very cool paper that got quite a bit of attention. And what we did there is try to basically find a way to detect senescent cells through what we call the surfaceome, so the pattern of surface proteins in the senescent cell. We designed these sort of nanoparticles that would bind to one of these epitopes on the senescent cell and would deliver a toxic drug, a toxic cargo like this, or perhaps even a fluorescent marker to identify senescent cells. And again, this is we're not reinventing the wheel here. We're just trying to use the same thing that's been used in cancer. There's many success stories with monoclonal antibodies targeting cancer cells through the surface epitopes. We're trying to do the same, but changing the epitope and using these sort of markers of senescent cells. And the first thing that we did is to publish a screening of the senescent cell surfaceome. Was the first screening of the surfaceome, and now people are using those epitopes to design these targeted senolytics. So the first one was these nanoparticles, and here's a picture, a blow-up picture of these nanoparticles that are sort of generated. Almost the idea is to you stick an epitope and you have these polymers form around the epitope. When you remove the epitope, the polymers act like antibodies. Basically, they have this sort of imprint of the epitope you wanted to target. When you use this in cells, and let's start with the bottom figure. Here, what you actually see, the shape of the cell here is actually highlighted by the green fluorescent protein in these nanoparticles. These are nanoparticles tagged with a fluorescent protein, and you can see they're stuck to the membrane of senescent cells pretty nicely. So they seem to be selectively recognizing this epitope on senescent cells, and you can see this in a less blow up picture. You can see when they become senescent, these cells accumulate a lot of these nanoparticles that are tagged with a fluorescent dye, and they're imprinted towards this epitope. And there's some background, you can see some background noise, but there is sufficient difference to actually allow us to kill the cells. And this is what we did here. So if you change the fluorescent dye for a drug, what you can achieve then is killing of these cells. In this case, we use the Ibrutinib, and this is a drug that on its own doesn't kill the cells. You see the black bar is the control cells, the gray bar is the senescent cells. With the Ibrutinib, they don't die. But when we actually deliver the Ibrutinib with a nanoparticle inside specifically these cells, we see quite a significant difference between the control and the senescent cells. So what we show here is specific delivery of a drug into a senescent cell that can be used to target senescent and kill senescent cells. We tried it in vivo, but not the killing one. We're still working on that. We needed to get the right permissions for that. In the UK, working with animals is not so easy. You have to ask permission for anything. Actually, working with all animals is difficult because we cannot let them die of old age. We have to call them before, because being old is something is considered too much of a stress for mice. So it makes our lives quite difficult. So, but at least we managed to use these nanoparticles tagged with the fluorescent protein to actually see if you compare here, and this is the quantitation of that, in old animals, you see many more of these nanoparticles accumulated, mostly in the abdominal cavity, and we still don't understand why, because it should perhaps be more equally distributed. But there is a difference in the old animals compared to the young animals. And again, here with these nanoparticles given orally, and we tried the different ways of delivering these nanoparticles, they all had similar effects. So we designed a nanoparticle that can detect senescent cells. Now, again, would this work for aging or age-related diseases? Would this work for cancers? Would this be a way to clear senescent cells after, for instance, chemo or radiotherapy to actually prevent this relapse? I'm thinking mostly on solid tumors, but would that work also on leukemia? That's one of the questions that we have. And this paper we published last week, so it's a very new paper, and you may have seen some of the press releases that's been in the news this week about this paper, that we took this one step forward, and we designed what we call an antibody drug conjugate. Same principle. The idea is to recognize an epitope on the surface of senescent cells, tag it with an antibody, tag it with a toxic drug, and then have this antibody being internalized by the senescent cell and deliver this drug inside. Now, the advantage of this compared to nanoparticles, that nanoparticles are very cool and they look very nice on papers, but they're never going to let us put a nanoparticle into a human yet, at least. We're very far away from the clinical applications of nanoparticles. We can put them in animals. We probably cannot use them in humans just yet, mostly because we don't know what's going to happen once you have all these sort of really small molecules going through your bloodstream. One of the fears is that it's going to get stuck in the kidneys, and they're not going to be able to. They're just going to cause a renal failure because they're not going to be able to be cleared. But antibody drug conjugates, that's something that's already been used, and again, this is being used in cancer. So we're trying to use the same idea. So we are using an antibody against one of these targets of the surface, and we design this antibody drug conjugate, as you can see here. The key to this is to have the right antibody for the right target, and then link it with this molecule, a linker, to a drug. And you could have the drug of choice. Here we use duocarmazine, which is a drug that's being used quite often in cancer for antibody drug conjugates. And the key is that the linker has to be of a certain chemical structure, so when it enters the cell, it's cleaved. And the antibody, I don't have the figure right now here, but the antibody, we saw that it gets internalized in the senescent cell, and then the linker would be cleaved, this drug would be released, and the cell would die. And just to cut the long story short, this is the paper that we just published, if you are interested. You would see that the difference, the therapeutic window, let's say, of control versus senescent cells in different models that we tried is quite big. If you, for instance, compare these blue lines or the green lines, which is the positive control, there is a difference in cell viability treated with this. Did I press something wrong? Right. So there is a significant difference, a statistically significant difference in cell death induced by these antibodies in senescent cells. Through, again, the marker we use in this case, the epitope is something called B2M, which you probably know is it's a protein that it's expressed in other cells as well, but it's highly expressed in senescent cells. It may not be the final target when we think about the clinical application for this, but we have many other targets in that surfaceome that we found. So this is just a proof of principle that this can be done, that we can design antibodies, we can tag them with drugs, and they would have a differential killing in senescent cells. Now, the next step would be to refine this, have even a better window here, try it in vivo in animals, and so on. Again, this is very expensive experiments, designing these antibodies, so that's why we had to stop before putting them in animals. We ran out of the antibody, basically, and we hope to be able to follow this up soon. Right, so this is what we're trying to do. So try to design these sort of smart bombs, whether it's nanoparticles, antibodies, something that could kill senescent cells, for many reasons, because this we think could help age-related diseases. This we think could help cancer, and could be perhaps one of the things that would reduce the possibility of relapse. So just to summarize what I said today, so at least in the field of leukemia and cancer in the lab, we're quite focused on this axis, how resistance and relapse appears. First of all, why some cells are already resistant, and why this resistance appears and leads to relapse. And what I show you today is that obviously there's a lot of things that we think can influence this axis. chief among them probably mutations that are selected through exposure to a drug. But I hope I convince you, or at least give you the idea that there's other factors that could be playing a role, like p53, like the presence of perhaps senescent cells, like the levels of reactive oxygen species and how they are buffered in the cells. This may be influencing this axis and helping all the different things that happen in the cells to actually cause this. And on the other side are the things that we can do about it. We can try to find better personalized medicines, probably combined with other things. So combination therapy is probably the way to go forward in personalized medicine. I'm a big fan of the idea of repurposing drugs once you find the right mutation in these cells, and things that could perhaps be perhaps not the choice or the first thing that you would think of, like adjuvants that could help, like senolytics, these drugs that could, for instance, get rid of senescent cells. Again, this is something that in epithelial cells, epithelial cancers, we know it works. Now we're trying to see whether this is something that could happen as well in B-cell malignancies, and let's see if we can in any way help improve the treatment of these diseases. And that's everything I wanted to show you today. This is just the team that did this work. Actually, this is the last picture we took before lockdown, so it's a bit outdated now. We haven't been able to get all of us back together. So some of them left already. Some of them, there's new ones that came. And this is the work we do in the lab in Leicester, and these are sort of our funders and our collaborations in the Dyer lab. And thank you very much for listening. I'll be happy to take any questions that you may have.
Thank you, Salvador. So questions?
First of all, congratulations for the nice talks. And I wonder when you present first the history about P53. That was really nice that you showed that the phosphorylation in serine 63, if I don't remember well, was specific for to understand what is happening in the cell lines, in the leukemia cell lines. But I wonder why you don't use the genome enrichment to detect more molecule that can help you to understand exactly what is happening in that cells.
Absolutely, yes. No, that's the next experiment, and it's the way to go. As you can see, some of these experiments are quite preliminary, and they quite sort of low tech approach. So instead of doing big screens, we went for candidate approaches, and these are projects that were, I guess, quite new for us as well. And before actually committing resources and money and time, we thought, okay, let's do a quick few experiments, see what happens. And we were successful in some of them, but I agree with you. I mean, to see the big picture here, we need to think bigger. And we are, I mean, asking for money for that. Obviously, that's the next step, generate some preliminary data. And again, we need to establish our name in the field of leukemia a bit before actually being able to get funding for this. And the next step would be to actually do something
okay,
like
good.
that.
We are open to collaborate. I am running the proteomics facility.
Let's talk about it. Yeah. So I'd be very interested in this. Actually, one of my goals is, and actually coming here, is to meet with you guys and start collaborations. We're very happy to do anything, try to establish collaborations in this. Here in Leicester, we can offer quite a lot of clinical samples. We have there, we have a good clinic, and we have a lot of clinical trials. We have especially a lot of interest in Btk inhibitors, and we're testing new things. So happy to do more research on that, yes.
Great job, thank you. I have a question regarding the relationship you mentioned between the BTK signaling and the phenotype of senescence and modulation of aging, basically under the treatment with the Ibrutinib. So do you know whether this phenotype is the control by Ibrutinib is strictly dependent on BTK, or it could be related to other non-BTK kinases affected by the drug?
Yeah, absolutely. Yeah.
And the second question is, do you see the same phenotype using another BTK inhibitor like reversible inhibitor or something like that?
Yes, when we did this, we tried a couple of BTK inhibitors. and we're testing now on other ones. I agree with you. These kinase inhibitors are dirty inhibitors, even if they are supposed to be inhibiting one thing, they're inhibiting many other things. What we tried in the original study, we confirmed that BTK is involved in this pathway by also genetically reducing the expression of BTK by shRNA and so on. So there is quite a specific BTK effect on senescence. Now, obviously, in vivo, this is a bit more complicated to do. I mean, we cannot just do this sort of experiment so easily. And when we are treating these animals with Ibrutinib, we are doing many things at the same time. And I agree with you. And so what exactly is the effect on aging that we see of Ibrutinib, how much of it is through BTK, how much of it is through some additional things, we can't say for sure. What we know is that in vitro, yes, in vitro there's a clear dependence of senescence and BTK. Now, if BTK inhibitors are also contributing, for instance, to lowering sort of a background chronic inflammation that you see in aging, perhaps that is also helping through another kinase pathway that is inhibited by BTK inhibitors. We don't know for sure. Yeah, I mean, I think there's a lot of questions still to be answered there.
Hi, here. Thank you for the talk. So my questions are also related to the first part, the p53 one. So you're detecting phosphorylation at the serine 15, which is usually associated to DNA damage. So have you also evaluated DNA damage in the same conditions as you detect the ROS?
So yes, I think we think that potentially that's the explanation for this phosphorylation. So that the fact that you put these cells in this sort of microenvironment in which they're supposed to be happy, but the fact that this microenvironment generates ROS, that is triggering some DNA damage signal that would lead to phosphorylation. So I mean, in different models we tried before, oxidative stress causes phosphorylation of serine 15. So we think, actually, when we blocked with antioxidants, we saw serine 15 disappearing. So that's, I mean, our hypothesis or our model is ROS inducing phosphorylation of p53 and increasing p53 levels, and the cell not dying is the part that's this disconnect here is the part that's difficult to explain, and we're still trying to
and it's connected to that. I think that there are some papers that show that 53 becomes acetylated in response to ROS.
Yeah,
maybe acetylation is also something to look for.
The postulation modification of p53 is a whole world, and if you read the paper on that, it's like they are this thick. I mean, because every day you find a new postulation modification. Acetylation, phosphorylation are the most important, but nowadays you can see anything sticking to p53. Now, how much that is important in function is something that is very hotly debated in the field of p53. Some people think that the function of p53, whether it's going to kill or not going to kill the cell, or arrest or not arrest the cell, depends on what phosphorylations or what post-translational modifications you have. Some other people think that that is secondary and there's other things helping. And for instance, in this case, could be, are we inducing high levels of anti-apoptotic proteins, BCL2 family, that even if you have p53, they would counterbalance that and block it? I think that's pretty much an open question.
Okay, thank you. Just a quick one. So sorry. Is it the P21? Is it normal? Because I know that the HAP1 is a chronic myeloid leukemia cell line. It has normal p53, but a mutation in P21. So I don't know if you have evaluated that.
Yeah, here they were normal in the samples that we saw. I mean, mutations in P21 are very rare because in cancers, because in most cancers, because P21 is definitely a CDK inhibitor. It's supposed to be a tumor, or at least in the tumor suppressor pathway of p53. But the problem is that some cancers actually like P21, because the fact that they have P21 up blocks, in a way, apoptosis or prevents apoptosis. So it's a bit of it's not a black or white thing, because if you have P21, the cells should be arrested, but having P21 protects you against apoptosis. So some cancer cells find a balance, and they prefer not to mutate P21, so they can use this sort of P21 function to their advantage. And in this case, we saw P21 was functional as well. It was normal in these cells, which contributes also to the confusion of why these cells are not arresting.
Thank you.
My part is on the senescence. Today you showed that you were looking for surface markers for senescent cells. Are there a marker for all senescent cells, or is a cell type specific?
That's a very, very good question, and that shows that you know the field of senescence. And the problem we have in senescence is that there's no way. Actually, let's rewind. So we were quite naive when we started the screen with this mass spec, this proteomic screen of the surfaceome. And we were quite naive, thinking that we would find a universal marker of senescence, which has been very elusive in the field. Everybody's looking for this, was looking for this sort of holy grail a marker of senescence, which now I think everybody agrees that it's not going to happen. So I think the consensus now in the field is that there's not such thing as a universal marker of senescence, and what we see is specific markers in specific situations. And what we're doing now as well is expanding our knowledge of the surfaceome in different conditions, because, for instance, P53-induced senescence would probably lead to one sort of surfaceome, P16-induced senescence perhaps to another one. Senescence of epithelial cells would have one feature. Perhaps B cells would have another feature of the surfaceome. So it's probably more heterogeneous and more difficult than we thought. Now, all this that I said of targeted therapies, basically, it's dependent on having a good marker. So what we're doing now is try to find the best marker for each condition. For instance, one of the things we're trying to do is try to find ways to get rid of senescent cells in lung fibrosis. It would be one of the most obvious indications, because lung fibrosis is an incurable disease. Idiopathic lung fibrosis happens a lot also in aging, and it's irreversible, and senescent cells play an important role there. So by looking at the surfaceome of these particular cells in this condition, we can design, hopefully, targeted drugs against that particular marker of senescence in that particular condition, and perhaps define a treatment in that condition. That doesn't mean that this antibody, if we use it for lung fibrosis, is going to work for Alzheimer's or for any other disease with senescent cells. Probably we'll have to look for different markers there that would maybe be in the same list, or perhaps if you look at the whole profile of the surfaceome, we may have some coincident markers in some situations, but not anything probably that is transversally equal and present in all the senescent cells.
It's just because the beta-2 microglobulin is high in MHC1. So.
Yeah, it's everywhere. I know it's supposed to be expressed in many cells, and that's one of the things that we say it's going to have a lot of background, and if we design a drug against this, maybe killing a lot of other cells. But we're not the only ones now. We're the first ones defining this marker of senescence. There's been other people working on this marker. Seems to be one of the ones with best response in some models. How is that going to work on a clinical setting? We don't know. Last week I started talking with a Japanese company that are interested in this antibody for treatment of oculopathies that are basically aging in the eye. Whether that's going to work or not, perhaps that's the setting. In the eye, perhaps you're not going to have that much B2M expressed. If you do it systemically, maybe yes, you're going to kill everything. Who knows? Is there enough difference in our cell models? Yes, there's a huge difference in senescent cells. They express much more B2M than the non-senescent cells. But is that something that if you do it in an animal? Again, none of these targeted therapies have been yet given to animals, so we don't know if that would perhaps kill other cells. The advantage is that we have a huge list of markers. So if this one doesn't work, we go to the next one, and we find it for different conditions. It may be a good marker, a good target in particular conditions, maybe not the right one in others. But yeah, this is a concern that I share, yes. Thank you very much.